We are now starting to get into the fun genetic stuff. But first, we have to learn about this process of meiosis, because in the last chapter we learned about mitosis, now we have to learn about meiosis. So remember from last time, we learned about the egg and sperm cells. We said these are the gametes, which means these are the sex cells, and these two types of cells are haploid. So for humans, that means that each one is going to have 23 chromosomes. When the sperm fertilizes the egg through fertilization, it produces the first cell, which we said is a zygote. This is the first diploid cell, meaning this one is going to have 46 chromosomes. Diploid cells are then going to reproduce and divide through mitosis, and that's going to form those specialized cells like skin cells, stomach cells, blood cells, whatever. And these cells all divide through mitosis. Now, the egg and the sperm cells, though, they do not divide by mitosis. They divide by meiosis. So meiosis is basically egg and sperm cell division. Now, this is going to be important later. Remember that we can make these maps called karyotypes that will show you the pairs of chromosomes. In humans, we said there's 23 pairs, but each person has 2 copies of each, so there's a total of 46. So for example, in humans, set 1 through 22 are your autosomes, which give you your basic characteristics, whereas the 23rd pair is the sex chromosomes. Every organism has a different number of chromosomes. So for example, chimpanzee, which is pretty closely related to humans, but chimps have 24 sets of chromosomes. They have an extra set. 1 through 23 are their autosomes, and then the 24th set is their sex chromosome. So while humans have 46 total, chimps have 48 total chromosomes. Here's a chart I found online just showing you some other examples. Fruit flies are very small. They only have 8 chromosomes. Ducks have 80 chromosomes. Watermelon has 22 chromosomes. So every organism is different. Now, if I had to draw a human cell for all my examples, I would have to draw 46 chromosomes in it. It would take me all day to draw all of these chromosomes, and you guys would be falling asleep just bored out of your mind. So for my examples today, I'm going to use a fruit fly, because then I only have to draw 8 chromosomes. It's a lot more doable. But remember, everything that I'm drawing today for a fruit fly also applies to a human. Okay, everything I'm drawing for the fruit fly applies to the human. I'm just drawing fruit fly cells because I don't feel like drawing 46 chromosomes every time. Okay, so let's say we have an egg cell. We'll make the egg green here. Remember, egg cells are gametes, so they are going to only have half the number of chromosomes. So for a fruit fly, an egg cell is only going to have 4 chromosomes. Now we'll make our sperm cell purple. Don't forget, sperm cells are also gametes, so they're also going to have 4 chromosomes. Now, when this egg and sperm come together, they're going to form a zygote, a fertilized egg cell, which is basically the first cell. This zygote is going to get all 4 chromosomes from the egg, and it's going to get all 4 chromosomes from the sperm. So in the end, this zygote has a total of 8 chromosomes. Now, I drew these chromosomes so that they look like they're lined up together. Each set is lined up. That's because chromosome set 1, okay, has 1 chromosome from the egg, 1 from the sperm. So this is the first set. The egg and the sperm both have similar genes that are lined up next to each other. So we'll just say if this is a fruit fly, there could be a wing gene here on this egg chromosome, and the wing size gene for the sperm is going to line up with it. Maybe on the second set of chromosomes is the eye color. I'm just making this up. So maybe the eye color gene is somewhere up here on this one, and it's also here. They're going to be lined up. So later we're going to talk about which gene is going to basically win over the other, which one's dominant, which one's recessive. But in all beings, there's 2 copies of each gene, one that came from the egg and one that came from the sperm. So there's 8 chromosomes, but each one is a set. And because these genes are very similar to one another, we say they are homologous chromosomes. Homologous means similar. They both contain similar sets of genes. Now, when this zygote divides into cells like skin cells or wing cells or eye cells or whatever, these cells are going to divide by mitosis. And as you know, in mitosis, one cell simply splits to two. And you can see both of these two new daughter cells are identical to the parent, and they're also identical to each other. So again, in mitosis, all cells identical. And also notice after mitosis, all of the daughter cells have 8 chromosomes just like the parent. So they have the full diploid number of chromosomes. So that's basically a review of mitosis. And again, these are somatic cells like skin cells, wing cells, or whatever. If this were a human, it would be exactly the same, except for instead of only 4 of these little ones, there'd be 23 over here, 23 over here, and all of these new cells would have 46. But it would take me all day to draw that, so I'm just drawing you a fruit fly as an example. Now we reviewed what happens during mitosis. Now let's look at what happens during meiosis. So again, for meiosis, we're starting with a parent cell. This parent cell is diploid, which means if we're talking about a fruit fly, we're starting with 8 chromosomes here. Now, if this cell, we'll pretend this is going to be turned into sperm cells. So this parent cell is the parent of future sperm. Now, the first thing that's going to happen before these cells go into meiosis is the chromosomes are going to double themselves. So it's going to look like this. The original cell had a single chromosome, 8 of them total. And then they double and they form these little X-looking things. You could think maybe now it went from 8 to 16. But we no longer call these little lines, we no longer call them chromosomes. We now call them chromatids. So we went from 8 chromatids to 16 chromatids. Each one of these chromatids is held together by what I made these little red dots. And these little red dots that are holding the chromatids together, these are called centromeres. What's going to happen next is all of these chromatids are going to line up, but it's going to be like each set will line up with each other. So it would look something like this when they all line up. Now, if the cells were just to divide down the middle right now, that would be kind of weird because that would mean one cell gets all of the same genes that originally came from the egg, and the other cell would get all the original genes that came from the sperm. And that wouldn't be right because that would mean that you would be identical to one of your grandparents. And that's just not possible, right? You don't look identical to your grandparents. So at this stage, something super amazing happens. Yeah, this is literally like the amazing step that's coming up right here, okay? Super amazing. So what happens during this amazing step is something called crossing over. So here's what happens during crossing over. If you zoom in on one of these sets of chromosomes, they originally look like this. But what's going to happen is some of the parts from this chromosome are going to swap with parts from this one. So what you're going to end up with is chromosomes that are all mixed up. So some of the genes from this one went over here, some of the genes from this one go over here, and they're all mixed up all over the place. And this is what introduces that variation. This is why 2 siblings, unless they're identical twins, will never be identical. You could never be identical to one of your siblings because of this step, crossing over, that happens in meiosis. So where we're at now is we still have this one single cell that has 16 chromatids, but all of the chromatids are all swapped up and mixed up. So we had crossing over already. And each one has a set that's lined up. Set 1's lined up here, set 2's lined up here. Now what's going to happen is a series of prophase, metaphase, anaphase, and telophase, another set of PMAT. After the first PMAT, we're going to end up with 2 new daughter cells. Now, each one of these daughter cells has 8 chromatids because they were divided in half down the middle. So half of the cell's chromatids went to one cell, half went to another cell. And don't forget, each one of these chromatids is different because there was swapping, crossing over. One more thing's going to happen during meiosis. These cells are going to divide again. Each one of these is going to divide into 2 new cells. It's going to do this by going through prophase, metaphase, anaphase, and telophase a second time. So this is PMAT part 2. The first one was PMAT part 1. This time when the cells divide, we're going to have that spindle thing come in, like the one we saw before during mitosis, the spindle is going to pull these chromatids apart. So right at the centromeres, they're going to get pulled apart. So in the end, we're going to end up with 4 sperm cells. I'll draw them like sperm. I'll give them little flagella. Each one of these sperm cells has 4 chromatids total. So we started with 8 and now it's chopped in half to 4. And each one of these sperm cells is different than the other. There's genetic variation. So from starting 1 cell, it went to 4. Any one of these sperm cells could fertilize an egg, create a zygote, but only one of them could, meaning this sperm cell would create a child that looks very different than any of these other sperm cells because each one has different genes located in their chromosomes, different sets. So overall, if we were going to compare mitosis with meiosis, in mitosis, we're going to start with 1 cell and we're going to end with 2 cells. In meiosis, we're going to start with 1 cell and we're going to end with 4 cells. In mitosis, the daughter cells are identical to the parent. In meiosis, the new daughter cells are genetically different from the parent. There's genetic variation. In mitosis, we go through 1 cycle of prophase, metaphase, anaphase, and telophase. Whereas in meiosis, we go through 2 cycles of prophase, metaphase, anaphase, and telophase. Mitosis is a way for somatic cells to divide, whereas meiosis is a way that gametes divide. So only the egg and the sperm. In mitosis, the parent cell was diploid and the daughter cells are diploid. Whereas in meiosis, the parent cell is diploid, but the daughter cells are all haploid. So meaning for a human, the parent cells started with 46 chromosomes. The daughter cells have 23. The last difference is in mitosis, there was no crossing over, which is that step where the genes all shuffle, whereas in meiosis, there is crossing over. And those are the main differences between mitosis and meiosis.